In this lesson, we want to consider the general chemistry of DNA synthesis and see how the challenge of the antiparallel nature of the double helix is met. The point on the DNA molecule at which synthesis begins is referred to as the origin of replication. As for most prokaryotes, the E. coli genome is closed, circular, double-stranded DNA. Strand separation begins at the origin of replication and is bidirectional. This means that two replication forks occur where new chains are formed, complementary to each of the two parent strands, as illustrated below. The bubble or eye that forms resembles the Greek letter theta. For prokaryotes, as shown in the figure to the right, there is one bubble associated with the one origin of replication. In eukaryotes, replication begins at multiple sites and therefore several bubbles or eyes are apparent. DNA polymerase is the enzyme that catalyzes formation of the phosphodiester bond in the polynucleotide structure. The new strand is synthesized in the 5' to 3' direction, which means that the last nucleotide added carries a free 3' OH group. The oxygen atom in the hydroxyl group serves as a nucleophile to attack the alpha phosphorus atom on the incoming nucleotide triphosphate. Formation of the bond results in the release of pyrophosphate, and subsequent hydrolysis of pyrophosphate makes this process energetically irreversible. Due to the antiparallel nature of the double helix, keep in mind that if synthesis occurs in the 5' to 3' direction, then the template must be read in the opposite orientation, that is, 3' to 5'. Now that the replication fork is formed, let's see how each of the two parent strands participates in the replication process. In the figure below, the parent strands are colored black and the daughter strands are in pink. The top parent strand is the 3' end. DNA polymerase can bind at that end and read the parent template left to right, synthesizing a new strand in the 5' to 3' direction continuously. This is referred to as the leading strand. However, we encounter a problem with the bottom parent strand, the 5' end. Recall that DNA polymerase must read the parent in the 3' to 5' direction in order to synthesize the daughter in the 5' to 3' direction. This means that DNA polymerase cannot bind to the 5' end and read left to right. Instead, it must bind upstream of the end and move right to left to read the parent and synthesize the complement. In essence, DNA polymerase is required to jump ahead to make a short fragment of 1,000 to 2,000 nucleotides long. Then it must jump ahead again, and so forth until the process is complete. Thus, synthesis of this strand is discontinuous, and it is therefore referred to as the lagging strand. The stretches of new DNA are called Okazaki fragments, after the scientist that discovered them. Now notice the two DNA polymerase enzymes bound to the two strands at the replication fork, pictured here as green spheres. This depicts the fact that the actual movement of the complex of DNA polymerase enzymes is indeed in one direction, left to right in this illustration. In order for this to occur, the lagging strand parent must loop out and wrap around in order to orient the template appropriately. The Okazaki fragments must ultimately be linked to form an intact DNA molecule by the enzyme DNA ligase. Having considered the overall process of DNA synthesis, let's next turn our attention to the enzymes that catalyze this amazingly complex process.